Oh hi! Today I will be sharing to you all about the art of protein synthesis. You might think that it's easy, but no, it's not. This is the most important process your cell goes through, because without it, you will not be you. So let's get started. Protein synthesis is a process that takes place in the cell of a living thing. And it actually consists of two processes: transcription and translation. In eukaryotic cells, transcription takes place in the nucleus.
Transcription is a transfer of genetic instructions from DNA to mRNA. During transcription, a strand of mRNA is made to complement a strand of DNA. Transcription begins when the enzyme RNA polymerase binds to a region of DNA to unwind, so the enzyme can read the bases of the DNA. The two strands of DNA are named based on whether they will be used as templates for RNA or not. The strand that is used as a template is called the template strand, which also can be called the antisense strand. The sequence of bases on the opposite strand of DNA is called the non-coding or sense strand. Once the DNA has opened and the RNA polymerase has attached, the RNA polymerase moves along the DNA, adding RNA nucleotides to the growing mRNA strands. The template strand of DNA is used to create mRNA through complementary base pairing. Once the mRNA strand is completed, it detaches from the DNA. The result is a strand of mRNA that is nearly identical to the coding strand of DNA, with the only difference being that DNA uses the base thymine and mRNA uses uracil in place of thymine.
But the new mRNA is not yet ready for translation. At this stage, it is called pre-mRNA, and it must go through more processing before it leaves the nucleus as mature mRNA. The processing may include splicing and editing, capping, and polyadenylation. Splicing and editing removes introns from mRNA. Introns are regions that do not code for proteins. The remaining mRNA consists only of regions called exons that code for the protein. Capping adds a methylated cap on the head of the mRNA. This cap protects the mRNA from breaking down and helps the ribosome to know where to bind to the mRNA. Polyadenylation adds a tail to the mRNA. The tail consists of a string of ACE, adenine bases. It signals the end of mRNA. It also involves in exporting the mRNA from the nucleus, and it protects mRNA from enzymes that might break it down. Translation. Translation is the second part of the process in which the genetic code in mRNA is read to make protein. Translation occurs in three stages: initiation, elongation, and termination. Initiation after transcription in the nucleus. The mRNA exits through a nuclear pore and enters the cytoplasm. At the region of the mRNA contains the methylated cap and the start codon. These are then joined by a tRNA, which contains the anticodons. After matching the start codon on the mRNA, this group of molecules, mRNA, ribosome, and tRNA, is called an initiation complex. Elongation. tRNA keeps bringing amino acids to the growing polypeptide according to complementary base pairing between the codons on the mRNA and the anticodons on the tRNA. As the tRNA moves into the ribosome, its amino acid is transferred to the growing polypeptide. Once this transfer is complete, the tRNA leaves the ribosome. The ribosome moves one codon length down the mRNA, and a new tRNA enters with its corresponding amino acid. This process repeats itself, and the polypeptide grows. Termination. At the end of the mRNA coding process is a stop codon, which will end the elongation stage. The stop codon doesn't call for a tRNA, but instead for a type of protein called a release factor. which will cause the entire complex mRNA, ribosome, tRNA, and polypeptide to break apart, releasing all of the components. Lastly, proteins travel to the Golgi apparatus within the cytoplasm to be modified for the specific job they will do. Thank you all for listening. I hope you all learned something today. If you did, please leave a like and subscribe. That will be greatly appreciated. And remember to always think outside the box.